I'm pleased to introduce and open this session on neuroimaging signals and their meaning. Up first is Uri Maoz, who's going to tell us a story about a spiking neural network model for slow ramping in the human brain. If you asked me to recommend a recent paper on spiking neural network models, it would be this one. So Uri, take it away.
Thank you. And I know you're very objective about that, as you're an author on that paper. So one good thing about being after a no coffee break is that if you fall asleep, I will blame the coffee and not my talk. The other thing I guess I have to say is, so I was here for the MPP conference last year, and I gave this very general kind of talk about why neuroscience and philosophy need each other. So this time I decided to go into something more technical. Hopefully it will be of interest. And the last apology is while I was not directly affected by all these fires, I was trying to get air purifiers and so on for our house in West LA was difficult. So if this talk is not where exactly I want it to be, I apologize. I had less time than I anticipated. Okay, enough of the apologies. So yeah, let's talk about gradual. Oh, okay. Not happy. Doesn't want to move. Let's try again. Still doesn't want to move. Interesting. What am I doing wrong? What did you? Arrows. Arrows. Oh, okay. Sorry. Yes, I have it on a laser pointer because if I put it on the other thing that's not a laser pointer, look, when I go here, it doesn't go here in the same place. So there is no representation. The mapping, the isomorphism doesn't work. Actually, right now, I'm in the bottom of my screen, and it's in the middle of your screen. So anyway, strange. But here it works, and then the arrows worked. We're good. Sorry about that. Gradual neural buildups. So gradual neural buildups have been known since at least the 1920s, and perhaps especially famous one is the Bereitschaftspotential, or in English, readiness potential, or RP, that was discovered in 1965 by Kornhuber and Dike. And it's an exciting discovery. The idea is this, that if you have a voluntary movement here at time 0, then leading up to that movement, you see, so in old EEG, up is negative, right? So you see ramp down, negative ramp, whichever way you want to think about it, ramp up, but you see this negative, this decrease in voltage, okay, above motor cortex. And I think that that's interesting because, as per its name, it was attributed to getting ready to move, to the preparation to move. And if before that discovery, the only way for you to know that I'm about to move would be to measure my muscle contraction, here you can supposedly at least go into my brain and go something like, look, he's about to move. We see something happening. And to some extent, a lot of the talk will be around that. So this RP, right, it's this gradual buildup that we see in the medial frontal lobe, and we can measure it with EEG, as I showed you, but also you can see it in magnetoencephalography with MEG. You can see a similar thing with single neuron recordings. You can even see something like that with fMRI. And more generally, I would say that the readiness potential, the RP, is really just an example of these gradual buildups that we see in the medial frontal cortex and in other parts of the brain. And we can see them again with various imaging modalities, and we can see them for various things. We can see it when you're telling somebody, get ready, and the go signal is going to come soon. That's right, the CNV. You can see it when you're kind of there waiting for a stimulus. Maybe you've done a task and you're waiting to see some feedback. Did you do it correctly or not? That's the SPN. and you can see it in motor planning, and you can see it in time perception and in other things. And in these various things, you can see it in humans, and you can see it in monkeys, and you can see it in rodents, and you can even see it, these are all mammals, you can see it even in invertebrates and crayfish. So this gradual buildup also occurs for other spontaneous behaviors that are not directly to do with voluntary action, right? With the, let's say, generation of creative ideas, right? Or if you make abstract decisions between two things that have nothing motor to do with them, at least not immediately. It could be if you're solving a problem, you have a eureka moment. It could be when you're trying to recall a memory and you manage to do it. It could even be when you have one image projected to each one of your eyes. You have this binocular rivalry, and when your perception moves from one to the other, you also see some kind of ramping activity. Now, more generally, of course, you have this also in EEG, in MEG, in LSD, and so on. You have this like 1/F property, right, spectral property, where the low frequencies are underrepresented in the signal. That is, to some extent, that also has to do with some signal changing more slowly than at least in white noise. So here I will focus on the RP, but really more as an example of this gradual buildup. And so let's talk a bit more about the RP. We already saw that it looks something like this, where this is movement onset. And you kind of notice that if you look backwards in time, there's this kind of like an exponential decay backwards in time. So if you ask, when does the RP start? There isn't a really clear answer. It's not that there's a moment exactly where it starts, because it's kind of like this exponential decay, right? So this is to say, but why does it matter when the RP starts? Well, the story is that the RP was well known already in the late 60s and 70s and the early 80s. There are hundreds of citations of the Kornhuber and D.Sc paper, but it really became much better known in 1983 with the famous, infamous, whichever way you want to call it, limit experiment that I'll go through briefly. The experiment is quite simple. The idea is that you sit there, and whenever you feel like it, you do some simple movement. Let's say raise your hand, or you can press a button or something. And at the same time, you look at a clock, and you're supposed to say, or you're supposed to remember when you first had the urge to move. So the clock moves. It would look something like this. You sit there, you go, when do I want to move? Okay, I want to move now. And then you say, well, the clock was, let's say, at 11 when I first had the urge to move. So this is the experiment. You do this again and again, trying not to fall asleep. And after that, at the same time, they measure EEG, and you can see this readiness potential we already know from Kornhuber and D.Sc. So then the famous limit result is, here's the readiness potential, movement onset. Let's say that the RP starts somewhere around here, but then when do people report having had the urge to move? Well, it's around here, which gives us this strange gap where supposedly there's the information already exists in the brain about the action. The brain maybe is getting ready to move, yet the person reports consciously making the decision to move only around here. So there's this strange gap, right, which led people to speculate that maybe the decision was made unconsciously, or maybe that even the conscious intentions have no part in this causal chain that leads to action. So in this respect, we care about when the readiness potential starts, right? Because it matters for the size of the gap, although most people wouldn't think that this readiness potential starts here or somewhere after you say that you've made the decision to move. So we care about it, but an even bigger question is, what does really the onset of the RP mean? Is that really the beginning of decision to move, right? So that is what limits thought, and to a great extent, for a long while, the field has thought. Although it's worth noting that James Ringold, already in 1985, I mean, he wasn't the only one, but he criticized it in a way that then later on, Aaron Schurger here made a whole model into that we'll talk about in a bit. But back to the original idea. The original idea is this, right? So the RP starts here. There's this unconscious interval, but then you only become conscious after you say, here, this is when I first had the urge to move. So here you become conscious of it. So then in this, if you want to call it early model or the RP, or the original model of the RP, onset of the RP is really the onset of when your brain prepares action. Okay? Maybe it's the onset of the unconscious vision or intention to move. So this is the original model of the RP, and let's now talk about an alternative model for the RP, and again, potentially for other gradual build-ups. So this model relies on two observations. One, how do we measure the RP? Well, we wait until the person moves, we or EEG, then we align it to when the person moves, and we look backward in time. We do this many times, and then on average, we see this nice ramp up the readiness over 40 trials, 100 trials, however many trials. So one is you always align to movement onset. The other thing is EEG signal, as we discussed, this 1 over F property, is autocorrelated, right? So we'll talk a bit more about autocorrelation in a moment. So what is autocorrelation? Well, broadly speaking, autocorrelation, a more autocorrelated signal remains similar to itself for longer, or you could say it's more stable, it changes more slowly. To some extent, it's more smooth, although smoothness is slightly differently defined. Let's take a look here. So we have signals here that this is actually the signal from the same random seed, but in a more and more autocorrelated fashion. So this is complete white noise, meaning the signal can be, the next point can be anywhere. There's previous point says absolutely nothing about where the next point would be, and the next sample would be. And then as things get more and more correlated, autocorrelated, you can see that the signal starts to move. I mean, it becomes more stable, right, if you will. You can look at what's called the autocorrelation plot. So this is how fast, like how stable the signal is. I mean, we don't really care about this thing, so it's just like how fast does the signal decay. Let's zoom in here for a moment, and we can see that this white noise decays immediately, while as we add more and more autocorrelation, it takes longer and longer for this to happen, meaning the signal stays similar to itself for longer, roughly speaking. Okay, so if that's autocorrelation, let's try to get some intuition about this model before we using just differential equations. So this is what's called an integration to bound model. The idea is that you start from a baseline, and then there is some kind of a drift through this space up to the time that you reach the threshold. So maybe this is how you go through space. And now, if you run this model many times, okay, in different colors here, and then every time you go to where you reach the threshold, and you look backwards in time, okay, then if you look backwards in time, then you would get some kind of an average signal. And the question is, what would it look like? So let's try to get some intuition about that. The average signal, so we start from here, and then let's say this is where we cross the threshold. So then what happens in the previous sample, the one just before crossing the threshold? So you could naively think, well, maybe it could be anywhere.
But if you think about it, it can't be here because once we get to the threshold, we stop. So we couldn't have been above the threshold before, right? And it can't really be far away from this because far away from this because of the autocorrelation, right? So things change slowly. So that means that the previous sample is somewhere here, and then to some extent you can use similar logic to say, well, the previous sample would be here, and then over time you would kind of try to make it back, you would get something like this, and this is indeed this is a model that Aaron Schurger developed and published in 2012 with colleagues, and I mean, this is the differential equation behind it, but he was really able to show that you can get the readiness potential from this kind of many drift diffusions up to a threshold. You stop, you see how things are back average, and you really get this. I mean, this is only flipped because the readiness potential is going downwards, but you would get you would get this shape here. So the alternative interpretation of the RP, this idea of the late intentions, suggests that the decision is really much later when you get to the threshold. Okay? And I mean, the conscious interval is perhaps a bit before that, depends on which version of the model you want to talk about. But the important thing for us is that the onset of the RP here doesn't really mean much. Okay, it does not mean that the decision has been made. It's kind of like the beginning of the trial or something like that. So in this model, the onset of the RP doesn't really mean that much. Now, let's talk about. I mean, this model was, of course, challenged, and I want to mention a specific challenge to this model that came from Patrick Haggard's group, work by with Pradeu and others. The task that they had here was, again, a task of whenever you feel like it, press a button. But there's always a but, right? But if while you're standing there and you decide whenever you feel like it to press a button, if, I mean, you get all these letters flashing in front of you, if there is an orange letter that flashes in front of you, then that's considered like a probe. You're interrupted by that. And then what you're supposed to do is, if you are already planning to move, if you are aware that you're planning to move, press the button immediately. That's the task. So if that letter, that orange letter comes and you are not aware, having prepared to move, ignore it. If you are aware that you're preparing to move, press the button immediately. Okay, so they found this RP-like activity that I'll show you in a moment when people reported the awareness to move. So if people that were not aware that they were about to move, and they didn't really see anything, but when they were aware, you get this. And it looks, again, this is, again, like the old way of up being negative, as you can see here. So this is a kind of readiness potential. And their conclusion was that that people are aware of their intention. Maybe they're like implicitly aware, but then when they're probed about it, they become like, oh yeah, I guess I was actually preparing to move. So they're aware relatively early, maybe earlier than when Libet thought they were. And this is related to the readiness potential, right? So the readiness potential seems to be specifically associated with this conscious experience. So, and this seems to pose a challenge to these stochastic fluctuation models, like Aaron's model, because those models predict that you'd become aware only, you'd even make the decision only when you reach the threshold, so you can't become aware of it early on because nothing has yet happened. So there is an issue, though, with their study that actually Jake Godinez, who was a postdoc at, move from the microphone, right? So is now a postdoc at Cedars-Sinai, was then a PhD student here, and he noticed that there's an issue with their design. I mean, it's kind of weird design, but he noticed a specific issue with it, which is that the participants in their design could be pressing the buttons for two different reasons. One is the reason they intended. They might be pressing the button to say, oh, I was getting ready to move, I'm aware of that, and the probe came and I pressed the button. But there could be another reason for them to press the button, which is they were getting ready to move, and when the probe came, it was too late for them to cancel. They couldn't anymore interrupt their movement, right? So it was beyond the point of no return. They had to press the button. And the problem with their design is we cannot tease apart these two situations. So we don't know whether this signal here is due to this or to this or to both. So then we designed a different study. When I say we, it's the royal we. Jake did all the work. I'm taking all, well, not taking all the credit, but I'm standing here and telling you about it. But anyway, so we did this study, and this time we again asked people to do self-paced button presses, and they were again randomly interrupted by probes. This time the probes were auditory probes because we can better temporally discern auditory probes. And we also gave them a different instruction. If they heard the probe and they were preparing to move and they should inhibit the movement. And that's different from the study, right, the 2019 study by Elizabeth and Patrick. And it is, though, similar to the original probe study that was done already in 2006 by Shibasaki and Habit. So after they move, we then ask them, were they preparing to move? So let me walk you through this. People are told not to move for the first 2.5 seconds in this task, and then after that, there's a certain distribution, gamma distribution that probe comes at, and there could be trials where the probe just didn't arrive by the time you move. No problem, that's just a movement trial. There could also be error trials where you move too early. We actually don't care that much about this, but we do care about these other two conditions that happen, which is that the probe arrived and you did not move. If the probe arrived and you did not move, we ask you, were you preparing to move? And you can say yes, no, or unsure. People very rarely said unsure, so we'll focus on the yes and the no. And in our design, importantly, we can tell when people couldn't stop themselves from moving, when they failed to inhibit their movement, because we see the probe, and then they still move. And usually, of course, that happens. They move almost immediately after the probe when this happens. So when people move, there's the failure to inhibit. When they didn't move, there's the preparation to move. They say yes or no. And both of these together are what Perez Bucharaz et al. Called their awareness trial. So now, what happens if we look at the EEG? Well, here in green, you can see the people when they move, so you get an RP of sorts. When they couldn't inhibit, so when they failed to inhibit, you also see something like a readiness potential. But then actually, we did not see any difference when people were preparing or saying that they prepared to move or not. We really didn't see any difference here between the blue and the red. And the statistics bears this out. And if we now artificially recreate a signal that Perez Bucharaz et al. That they recorded, right? So if we add the failure to inhibit trials to the preparation trials, we get this signal here. And now let's flip it so we can look at them next to each other. So now we show voltages in the other direction, and you can see how similar it is to this thing that they got here. So we concluded from our study, and it's now forthcoming in Imaging Neuroscience. It should be out soon-ish, I think, right? So this RP-like activity that we see, it seems to be, that we see in Pradeu seems to be because of this combination of the failure to inhibit trials and the preparation trials. And yeah, and then what we see, what they see as evidence against the early decision models, we think is therefore less compelling, right? And it's not really much of a challenge. So the last thing I want to talk about is actually go back to these slow fluctuations a bit more, because we already mentioned that these stochastic fluctuation models, right, they require autocorrelated input signals that trend towards the threshold, and then there's the back averaging. But there's the question of where does this autocorrelation comes from? And put differently, if the autocorrelation in the input signal is too low, if there isn't enough autocorrelation, the model is not going to like slowly ramp up. It's just going to kind of jump up, and we'll actually see that in a minute. So we need these slow, we need this autocorrelated signal. And I mean, you can ask, first of all, I mean, well, do we see this autocorrelated signal in the relevant brain regions? And I mean, the answer is yes. This is from Schmid et al. 2011 paper. You can see these are single neurons. If you average them across a few trials, you can see this nice ramp up over the span of seconds, right? So we see this. But then, if we look at the autocorrelation functions of single neurons, we see something that doesn't really square with that initially, at least, because their autocorrelation decreases to very low levels, to near zero, within a few hundred milliseconds. So we have neurons whose autocorrelations decrease to zero within a few hundred milliseconds here, and then, but we have overall these ramp ups, and it's not really clear how these ramp ups can be from these neurons that are really that have these very short autocorrelations. And of course, the answer needs to be that it's somehow a network. So really, the mechanisms that underlie this emergence of these low ramp ups from these single neurons that have short autocorrelations was not well understood. What kind of network phenomenon would make something like this happen is what will the essence of the study that I'm going to describe. So what we did here is and again, this is a while we did much of the work here, and it's combining spiking neural network simulations with the actual data from Schmid et al. That they kindly let us have, and we were trying to understand this mechanism that creates this these ramp ups from these neurons with short autocorrelations, and we were able to find a spiking neural network that both created spontaneous fluctuations. It didn't. The fluctuations were not because of some input; they were spontaneous, and it created this slow ramping activity. And we in a task that's similar to the limit task, and I'll show you what I mean in a moment. So let's talk about the architecture of this model. So this network consisted. This is a spiking neural network model. So going back to Jay's talk here, right? This is really a spiking neural network. It's not a ReLU or something like that. And we have 400 neurons. They're leaky, integrated, fire. 80% of them are excitatory, 20% inhibitory, and they're grouped into 4 clusters. And the first thing to understand here is that there's something called the clustering degree, which is for each neuron, how much do they their cluster versus to other clusters. And note here that there's this inhibitory population here, means that there's inhibition going on, but there's also excitation. So it has to do with the two. So when you're just as likely to send an excitatory projection to within your cluster as you are to outside the cluster, then this REE, the clustering degree, is 1. And then in this situation, you do get this asynchronous spiking, but you certainly do not get low spontaneous fluctuations. So this is one extreme. At the other extreme, if you're basically only sending projections to your own cluster, then you get the classical winner-take-all. So then when one cluster starts ramping up, it just kills off all the other clusters, suppresses them completely. But then there is a regime in the middle where you're more likely to send projections to your own cluster, but you can also, and you do send projections to other clusters, excitatory projections. And again, you get this inhibitory pool that it inhibits everything. So when this REE is somewhere between 2 and 5, then you get, as we'll show, you get slow spontaneous fluctuations. And you get this nice combination of inhibition and excitation between the clusters that does what we want. Another element that we put into the network is slow synapses. So before we did this work, people have played with fast synapses, so synapses that integrate over 5 to 10 milliseconds. They integrate over the input, but we were looking at slow synapses with, the rise and fall of around 100, the rise around 20, the fall around 100. So this is much longer time. So let's see what the network dynamics looks like, and we'll start with a fast synapse, and you can see this like asynchronous fluctuations. Things are all over the place, and then as the REE increases, right, we can see that we get what's called this multi-stable or quasi-winner-take-all. So quasi-winner-take-all means that once one cluster starts being higher, it somewhat kills off the other clusters. So it comes at the expense of others. So this is for fast synapses. For the slow synapses, you actually see things that we were looking for, see these low ramp-ups, right? And again, if one cluster ramps up, the other clusters ramp down. You can here, if you want to see, you can compare the two, and you can see the difference between the slow synapses. And this is not only slow, so 50% slow, 50% fast compared to only fast here. So let's look at what happens to neurons in this simulation. So we defined here a threshold, and the threshold was defined for the entire cluster. That means that the average activity at a cluster had to be above a certain threshold for us to stop and call it movement onset. So if we stopped at movement onset and look backward, we can see that for the fast synapses, we get this really fast, right, ramp up. But for the slow synapses, we get this kind of ramp up that we were hoping to see. And you can see it ramp up or ramp down depending on the type of neurons. And I should say that this upward and downward neurons is the same thing that you see in the human brain as well. And if you compare this to actual neurons that we see in Schmid et al., you can see that there's a relatively good match, especially for the slow, the 50% fast, 50% slow. So this is what we call here slow synapses. You can see that they match quite well with what we see in the human brain. Not so much the fast ones. Actually, if you really do an R-square analysis, you can see the slow ones, the R-square is around 0.7, and for the fast ones, it's around 0.3. And really, our model was more focused on the slow synapses. It's also worth noting that the slow synapses that this is a gradual process. So if you only have fast synapses, you get this, and then as you introduce more and more slow synapses, you get more and more of this slow ramping. We started with this, but let's go back to this and say, if you look both in the model and in the actual neurons, if you look at the autocorrelation function, you can see that really the decay is quite quick. I mean, within a few hundred milliseconds, you're at zero, yet you get these ramp-ups that go for more than 2 seconds. So we were able to replicate the key phenomenon that we were trying to replicate, which is create a network phenomenon of these slow ramp-ups from units that have short autocorrelations. Another thing to note is that if you look on single trials, if you see these raster plots, I mean, it's hard to see here. Maybe it's a bit easier to see here. If you look at these raster plots, like on every trial, it's a mess. There's a lot of variability, but on average, you can see both in the simulated network and in the empirical neurons, you can see that there's a ramp up or a ramp down, depending on the type of neuron. But on every single trial, and here these are more examples, on every single trial, there's a lot of variability. Okay. So I guess if you look at those neurons that are involved in the network, we see a lot of variability. The question is, well, why? Well, the answer is, remember that the network gets to the threshold when, on average, right, its activity is beyond the threshold. So on average, that's what we see, but then each individual neuron can jerk around and be more variable. Right. So let's look a bit at the slow ramping at the population level here as well. It's interesting to look at if we try to classify between the baseline. So the baseline is, let's say, before this even starts here. Okay, the baseline and the activity here. So if you expect a ramp up, you expect to be able to classify better and better as the ramping activity gets further and further away from the baseline, which is, and you see that for the slow ramping as you would expect. For the fast ramping, because there isn't much happening until the very end, right? Then you're not able to really classify much until the very end. And you can see that, generally speaking, the slow increase in the classification ability is more similar to what we see in the real neurons here. So let's just take an example. Let's look at AUC, so roughly the accuracy of 0.75. We can see that for the fast neurons, they reach it very late, very close to movement onset, while for the slow neurons in the network, they reach it earlier. And again, for the empirical data, you can see that you also reach it earlier. It's also worth noting here that for the simulated data, right, you can see that there's this jump up here when these neurons, the fast neurons, start getting engaged. It's not surprising. And here you can actually see that the network becomes even more predictable, or more classifiable, more predictable, if you will, or more differentiable when the fast neurons click in. And as I mentioned before, our model doesn't really model these neurons that much. So in that, I'm also with Jay. This is a model. It's not the model. It's not the model to end all models, and so on. So another thing that we looked at is this: if you have a ramp up, there are certain things you would expect from classifying. You actually would expect classifiers to generalize into the future and not into the past, and I'll try to explain what I mean by this with this simple example. So let's say that we're trying to classify between this baseline and this baseline here and then this slightly elevated one. So the threshold would be somewhere here, which would then mean that this would be properly classified, but then all the other, right, all the other ramp, all the other activity that's up in the ramp would also be classified correctly as not baseline. But then let's imagine a different situation, and now we're trying to classify between the baseline and this very end of the ramp up. So you could imagine that the classifier, that the threshold would be somewhere here, or that the separation would be somewhere here, not the threshold somewhere. So now we would be able to classify these correctly, but not these. So we can see again, as I mentioned, that the classification generalizes into the future, right? As the ramp up continues, but not backwards into the past. And so if you train on one point, you should be able to test on any point in the future, but not in the past. And this is really what we see in the simulated data. And then if you slightly squint, you can also see it here in the empirical data. Yet one more point is just to say that we have an EEG proxy signal that we wanted to collect from the model. And what I mean by that is we know something about EEG. We know that it tends to reflect like the sum of the postsynaptic potentials below. At least I think we know this about EEG. So we can do that in our model, in our spiking model, and say, okay, what happens if we sum all the postsynaptic potentials? In this case, inhibitory and excitatory, although it doesn't really matter as we showed. Well, what do we get? Well, for the fast synapses, we get this really fast ramp up that doesn't look much like the RP, but for the slow neurons, we get this, right? We get something that really reminds you of the RP. It even goes in the right direction, if you will. And the reason for that is because if one cluster, why does it go downward? Okay, why is it trending downward? Because if one cluster wins, then the three other clusters, it suppresses them. So overall, the network gets less active. So if you sum up over the entire network, you're going to see a decrease, as we see here. And it's worth noting that this too is gradual, meaning that as we increase the percentage of slow synapses, you go from this gradually towards this, as you can see here, from the fast gradually to the slow. We did have this novel prediction here, but I think I'll skip that for time. But you'll just have to believe me that our model, just like any good model, was able to make a novel, non-trivial prediction about the network, about the neurons that was borne out by the actual data. And let me sum up by telling you a bit about what we found in this last study. So as I hopefully managed to explain, we demonstrated this spiking neural network that created these spontaneous fluctuations. And it's important to note here that these fluctuations that we see are not driven by any input to the network. It's just the ongoing dynamics of this network. We also got this, with the slow synaptic dynamics, we were able to create these slow, the slow fluctuations, these slow ramp-ups that I showed you. And we also showed that while the individual neurons in the network that had 50% slow synapses ramped up their firing rate slowly over more than 2 seconds, even though their autocorrelations were roughly order of magnitude lower. And this is just to say that the accuracy of the classifiers, we were able to go into the future but not into the past as we expected. This model also relatively well matched the actual data for individual neurons and on an aggregate. And I would just say here that, like, taken together, we can show that what we set out to show that there is, if able to get from low autocorrelation units a slow ramping as you'd expect. So some upshots, as far as we know, this is the first model to show this kind of slow ramping, especially for spontaneous activity. And another thing to say about it is these stochastic fluctuation models, the ones like the Schurger model, have been studied quite a bit, and this is an instantiation of those kinds of models in something that's much more biologically plausible and at a much lower level than those models. And one interesting bit here is in our model, depending on how you set the parameters, you could also potentially like go above the threshold, and then essentially at least after a while you could come back down, and you could potentially study cascades of decisions or cascades of actions or something like that, while most of the stochastic fluctuation models, once you get to the threshold, you just stop the model and you're only looking at one decision. So in our model, again, remember that the decision is not made until you get to the threshold, right? So there's no early decision in any way, even though you see these early ramp-ups. The early ramp-up does not really mean that the decision has been made. And in fact, also here, there's a paper by Luca and Aaron now that's a preprint that really shows that if you try to make a prediction about the upcoming movement, you can't do it until very, very close to an onset. The fact that there's a ramp-up does not mean that you can do PI based on something like that. I think of time's sake, I'll skip the future directions, and just to say that, I mean, this is a bit of an old picture of people at the PI, but while these are really the people that did the work, so Jake and Aaron were part of both the studies that I mentioned, and Uli Maoz from Cedars was involved in the second study that I mentioned. It's really, of course, the entire lab and the discussions in the lab and so on that made this possible. And I'll stop here and say thank you.
Questions?
Not everyone at once. Okay.
Unaccustomed as I am to asking questions. So I know this wasn't the theme of your talk today, really. You were talking about this mechanism, and I think the mechanism sounds great, sounds very interesting, sounds like it could explain all of this. I'm just, this question about a decision is one that I would like to ask about. I guess I feel myself as though, you know, I sometimes find myself, you know, I go bike riding frequently, and sometimes there's different routes that I can take, and I feel like I don't really know which route I'm going to take until I get to the intersection, and then I turn out to go one way and not the other. And I don't really feel like I make a decision. I feel like I'm finding out what my brain's decision was. And I'm just like, isn't that what's going on here? I mean, the brain is just spontaneously reaching this threshold, and then the action occurs, and I become aware that, you know, I'm about to press the button as I am actually starting to press the button. But I didn't make a decision. It emerged from the neural activity, and the behavior occurred, and the threshold crossing resulted in the action taking place. So is that how you think about it, or is this a different way of talking about it, or, you know, how do you guys think?
Well, first of all, this is not incommensurable with this model. This model doesn't necessarily claim that you have to make the decision early. Actually, this model is part of an idea that the decision is really made quite late, quite
But the notion that there's a decision sort of implies the existence of a decider who's a person looking at this and saying, oh, when I get to here, I'm going to decide to respond, as opposed to when I get to here, the action will be triggered through an outflow of activity through my nervous system.
So there are, okay, let me answer this way. In the end, if you get it to a T-junction, you're either going to make a right or a left. So something needs to happen, right? When you
I could stop. Occasionally I do stop, and, like especially,
there are three options.
Should I have taken this exit or not? I actually stop on that part of the exit where no cars are supposed to be, and I have to look at my cell phone and decide where I should have gone.
But when you were talking about a decider and so on, I think you're starting to talk about like, stop the world, I need to make a conscious decision here and let that conscious decision drive my actions or something like that. And that's what I'm saying that this model doesn't necessarily have. There isn't, I mean, this is part of, I think, the thing that we were trying to go against, this idea that maybe there's some kind of an early decision and you become aware of it really much later. I mean, in your example of the bike, in the end, when you get there, okay, you can stop. Maybe there are three options, but there are a limited number of options, and one of them you have to take, and that has to happen.
Yeah. But it's not, it's like we use the word decision because we're used to using the word decision and not because there's a decider. It's a mechanistic process that occurs when the threshold is reached.
But so when you ask how we think about this, well, I said that there are different types of situations. Okay. When you decided to come to this conference, we sent you an invitation, and I think you made a conscious decision. You looked at your schedule, you thought about, you thought about various things, and you made probably a deliberate decision to come. That's a different type of situation than when you get on your bike and you get to an intersection and you you just let yourself wander around. So I would say that there are different, for different situations, there might need to be different models. This again goes back to the discussion in the morning, right? There isn't one model to kill them all and in the darkness bind them or something like that. But I think that this tries to go against the notion that there was somehow, when you were riding your bike, there was some kind of an unconscious early decision.
Yeah, no, I'm totally with you on that. I'm not disagreeing at all about any of that. I think I understand that you're basically on the same page that I think I'm on. Of course, I'm not totally sure what I think.
That's okay. Neither do I. But
thank you. It's interesting.
I think Adina has her hand up. Is that okay? Aaron?
Yeah, so I'm just curious what the neural evidence is for these kinds of slow synapses, especially in the populations that you're interested in. Is this biologically realistic? Is this just sort of an engineering discovery?
So, as far as I remember, and maybe Jake, you remember all the details here, but I think, right, it's the AMPA, so there are these slower synapses, and as far as I know, they do, I mean, you're asking whether they would exist in places like the SMA, ACC, DLPFC, and so on. So as far as I remember, they do exist there, although, I mean, we didn't really talk that much about that in the paper, but I don't think that this is an implausible model in that sense. I'm not mistaken here, Jake. This is what I remember.
Yeah, that you got it. You got it.
Okay.
Uri, in one of your recent papers, that really caught my attention, you and your group showed that more sort of reason-based decisions, as opposed to this just when I get a whim, I'm going to press a button, aren't preceded by readiness potential at all. And I'm wondering how that fits into, all of the modeling that you've been doing with these diffusion models. Do you think for those kinds of decisions, are you just in an entirely different regime with entirely different neural substrates, or do you think this kind of stuff is going on in the background, but somehow gets averaged out. I mean, how do you fit, what's the big picture here with the two kinds of decisions?
Right. The work you're talking about was work with Liad Mudrik, Gideon Yaffe, and Christoph Koch. And what we think there is that the deliberate decisions, these big decisions, in that case it was donating $1,000 to a charity of your choice or something like that, we think that they're just made in a different part of the brain. So if you're recording over the SMA, you're not going to see them because they're just made, so the decision is made elsewhere. So then the SMA, you're just going to see something kind of flattish. So that's how we understand it. So it's really a matter of, again, going back to, I think that the discussion we had this morning about models is important. Like, what exactly are we modeling, right? We're not modeling the entire brain here, but so I think, but I still think that those, so, and we actually have some follow-up work that we're looking into that right now. So I think if we looked in the right part of the brain, we would see something like a ramp up, but it would not be the readiness potential in the SMA that you see.
Yeah, thanks. So it's just
it's just a different.
You hypothesize that it's completely different.
Yep, it's a different neural substrate. 531, I think.
Yeah.
Perfect.
I'll just add just one last remark to the discussion, is that the stochastic fluctuation model was designed precisely to explain these circumstances where you move for no reason at all. In other words, the slow drift of activity in your motor system is only going to show through in the average when it matters to the initiation of the movement, when it biases the precise time that you move. And that's going to tend to happen when you move for no reason, right? There's nothing left on which to base the time that you move, perhaps other than that. Maybe like when you reach that intersection. And so the model predicts that if you do have a good reason to move, you don't have that slow ramp. And that's exactly what Uri's data showed.
So